So good afternoon, students. Is my voice audible to you?
Yes, sir.
Excellent. Sorry, yesterday I don't know what happened, but both both the Zoom session as well as the website, Olympus Great Learning website, they both don't open. Although I had internet access, I don't know what happened. So I had to kind of upgrade my system software. Kind of working today. I don't know. I have to see. If not, I have to find another place. So hopefully it'll go all right today. So last class we finished 9.3, right? We have to start 9.4 now, correct? Did you happen to go through the content I sent to you on 9.3? Okay, so this is what I think best thing that you can do. Once you finish the class, go through the running notes and go through the given material so that you get a feel for it. You know, this is what normally happens when you're going through the class. Of course, multiple things are happening with you. So once you get a feel about the content given to you and the concepts that we discussed in the class, this is the first step towards your preparation. Once you get that done, right, once you're comfortable with the content given to you, once you know what actually is with you, because most of the students, what do? They actually gather the content, never get a chance to read it. And including me, I actually have more content than I usually read. So the problem here is we actually look for something that you already have. So instead, give it a quick glance. As I was telling you, you have to be really picky and extract the information from the content. Because for this anthropology, we have many sources where people have kind of went through and summarized it, but the essence of it requires for you to read at least some articles yourself, correct? But having no idea about content, I think it will be very, very difficult for you to do that. So the best thing here is understand the concepts in the class, read the given content to you, and see whether you are feeling comfortable with that or not. Wherever you are uncomfortable, that is where you have to raise the question. That is where you have to ask me, because by the time we finish moving on to this unit, like we're starting with 9.4 today, the best thing is finish reading the content at least once and ask questions on that. So you're comfortable with the content you have. So this is the first part. And then you get a confidence to review it later. When you go to the revision, you're still figuring out the conceptual understanding, then you will spend a lot of time and you freak out. Okay, that should not happen. Okay, so make sure to do that. Put enough time to read the content given to you. If not additional reading material, just cross-check what is extra given in there. Otherwise, the content I'm giving to you, okay, and your running notes. So make sure that you are comfortable with that. Okay, right. I know that we are kind of lagging behind on the answer writing part. Hopefully, we'll catch that up because I know last unit and 9.3 we finished, so we have to write some questions from that. So what we can do, I think for the, yes, 9.2 you have written. Evolution, I have to give you a few questions. The idea is same. I think we'll work on that question Yash was asking. Compare the Darwinism and synthetic theory of evolution. Take that question. So the Darwinism and synthetic theory of evolution comparison, I want. Okay, so that you actually can figure out both the Darwinism concept as well as synthetic theory. I want you to make a comparison. Okay, so I think I was briefly telling you on this. You work on, first explain what is Darwinism with an example, and then explain how synthetic theory is different from that. And expanding on the 4 points I've given while explaining both the theories is what is required. Right, Yash, you're asking this question to me, right? So work on this. This is a structure I want you to work on. Okay, and also I was talking to you about a video I made on Hardy-Weinberg equilibrium component. Okay, I'll come to that once we finish discussing about this. Right, what was the question? Yes, how Darwinism differs from the synthetic theory of organic evolution. So make a comparison on this. So where is the chat box? I would like to post this question. I'm pasting this question in the chat box. How Darwinism differs from the synthetic theory of organic evolution. So explain Darwinism. First thing that you have to do, okay, brief statement on what is evolution. What is evolution? Brief, very brief statement of what is evolution and what is Darwinian theory of evolution. Okay, Darwinian theory of evolution. Because only when you explain what is Darwinism, you can bring the comparison with the synthetic theory. So once you explain about Darwinism with key points, make sure to use the diagram I was showing you in the class. Okay, you draw that diagram while explaining Darwinism and use those key aspects of Darwinism. For sure, survival of the fittest, so competition, survival of the fittest, natural selection, these aspects you talk about. And then you start with the organic evolution. Here you have to use a comparative note. Till Darwinism, just explain as it is what is Darwinism. But when you start discussing about synthetic theory of evolution, start on the comparative note. So as compared to Darwinism, synthetic theory includes these following aspects. You talk about what is being added to the concept of Darwinism, because synthetic theory of evolution is a progressive concept. This developed over a period of time and it's an integrated concept of several ideas. So talk about those components that we discussed in the class. Where is variation coming from that Darwin could not tell. So even if you want, you can tell what were the shortcomings of the Darwinism and how synthetic theory of evolution complemented that. How synthetic theory of evolution added those components that were missing from the Darwinism. Okay. And talk about those components that we discussed in the synthetic theory of evolution. Okay. An integrated idea about that and how that is different, because you know it now. When you discuss about Darwinism, shortcomings of Darwinism, and how synthetic theory of evolution addressly tells synthetic theory has these components that were not part of the Darwinism. Okay. Okay. So next, I will, once you start working on this, I'll give you a few more questions. I'll give list of few questions from 9.2 and 9.3, couple of them, especially those that give you an idea about how to present information. Okay, we're not going to exhaustively work on every question here. Okay, so we have a list of questions with you. I give you a few selected questions. So those questions that would cover the concepts that are coming repetitively in the exam. Okay, and give you an opportunity for you to write. Okay, so today we are beginning with 9.4. Okay, unit 9.4, a very important chapter for the exam, because we do get frequent questions from this chapter and these questions are predictable questions. Okay, I'll tell you as we go through the content, I'll tell you what are those questions that usually appear in the exam. But there is, there are a list of questions that we will discuss about here, and mostly they are again repetitive in nature. Okay, I hope you had a look at the unit 9.4. So if you see the syllabus, 9.4 expects us to learn about the abnormalities in chromosomes. Okay, it asks us to learn about the abnormalities in the chromosomes. We already covered about the basics of chromosomes, chromosome structure, and cell division we discussed about, but sometimes cell division doesn't go as expected. There are few steps in cell division that go wrong, resulting in some abnormal number sometimes, and sometimes it is structural abnormality, meaning either number of chromosomes change or structure of the chromosome changes. So the idea is that chromosomes and chromosomal aberrations in man, they also mentioned the word methodology. Okay, so one component of it is numerical and structural aberrations. We first talk about numerical aberrations, structural aberrations, meaning number variations and variations in the structure of the chromosome. Second component, examples from them. Mainly examples from the numerical aberrations are given. Both B and C, if you see, if you read those names, 6 chromosomal aberrations, meaning abnormal aberrations refer to abnormalities. Abnormalities in the 6 chromosomes. How many 6 chromosomes we have normally? How many 6 chromosomes there are in females and males?
One pair.
Correct, one pair. Correct. So females have 2 X chromosomes, X and X. Males have 1 X and 1 Y chromosome. Correct. So chromosomal abnormalities happen on them, on those chromosomes, X chromosome and Y chromosome. Okay. So chromosomal abnormalities happen on them, and such 6 chromosomal numerical, these are number-based abnormalities, are Klinefelters. These are the names of the diseases we'll talk about today. Klinefelters, Turner syndrome, super females, intersex, and other syndromic disorders. Klinefelters, Turner, super females are some examples of them that we're going to talk about. This is part B. Part C is abnormalities in the not abnormalities in the autosomal chromosomes. So leaving out the pair of chromosomes, XX or XY, 22 pairs of chromosomes that we have, which are called autosomes, abnormalities in them. Okay, like Down syndrome. Down syndrome is also numerical aberration, number-based aberration. Just listen to me now, okay? You don't have to write much. When I'm presenting it, because this information should make it bit easier for you. This is like an intro part and understanding the syllabus. So autosomal means abnormalities in autosomal chromosomes, that is 22 chromosomes. Down syndrome is a number-based, number issue here. And Patau's Edward also numerical aberration. So change in chromosomes, chromosomal number results in these autosomal abnormalities, that is Downs, Patau's, Edward. And they also gave Cray-Duchat syndrome. Cray-Duchat is the only example for structural abnormality. Out of all these, in part B, 6 chromosomal abnormalities are all numerical aberrations. And in part C, all but Cray-Duchat syndrome are also numerical abnormalities. Only Cray-Duchat syndrome is a structural abnormality. So you need to know this. So what example to use for what? Okay, you'll understand this, but this is the idea. In part D, they have given some miscellaneous things related to chromosomal problems. What is it? So one is other way of causing genetic problems. That is called genetic imprints. We'll talk about genetic imprinting. Genetic screening. So how do you screen? In what stages do you screen humans? Okay, for genetic problems. And counseling. When do we need to seek help from counselors, genetic counselors, who tell us about the risk factors and help us understand the risk associated with a given disease and preparation towards a child like that, or making decisions like, not to have a child like that. So next to human DNA profiling, DNA fingerprinting, gene mapping, genomic study. There are over 5 different miscellaneous topics are there. Okay, we'll cover that in the end, but first focus is understanding what are chromosomal abnormalities, and next what are numerical abnormalities and structural abnormalities. Once I'll give you overview about that and what causes them, what causes such problems, then we'll focus on talking about the specific chromosomal abnormalities. Okay, did you understand what exactly we are going to learn as part of this? And once I'll cover a few questions on this, I'm going to go through the questions so that you understand what questions are being asked and what is expected from this unit. Okay. Right. Let's start with the abnormalities. What are chromosomal abnormalities? So do you remember what are the structural parts of chromosomes? If you have to draw structure of a chromosome, what parts do you see in a chromosome? Can you briefly recollect what parts we have seen in chromosomes?
Chromatin, DNA.
Chromatin. Okay. Chromatin. What is chromatin?
DNA chain, too many DNAs.
Chromatin is the semi-condensed genetic material is chromatin. Okay. So centromere. Do you remember centromere? What is centromere?
Middle part of chromosome.
Center part of the chromosome where two arms are attached. Any idea how are these arms called?
Sister chromatid.
Exactly. When are they called as sister chromatids? How many chromatids we have in a chromosome? How many such loops do you normally draw for a chromosome?
Loops.
How many? So usually you put a circle and you draw one like this. So this circle is the centromere part is a loop. So basically this is everything is same. It's one chromatid. So before DNA replication happens, right? Before cell division, we have one such chromatid, single chromatid. But whenever DNA replication takes place in the interface before cell division happens, right? There you write one more chromatid. So these two chromatids, which are copies of each other, because one acted like a template, another one is a copy of that. So these are called sister chromatids. So only after DNA replication, you draw two chromatids, but each of this, although it looks like four arms to us like that, it looks like four arms, okay? But literally, that whole thing is one chromatid, correct? Like that, two chromatids, sister chromatids we call. So chromosome has centromere. To centromere, two sister chromatids are attached after DNA replication. But before DNA replication, it's single chromatid. There's a constriction. It's like a loop, a knot, like a knot that is present in a chromosome. Ends of the chromosomes. How are the ends of the chromosomes called? The tips of the chromosome. Centromere is a central part of the chromosome. Tip of the chromosome. If you go to the tip region, corners of the chromatid, how are these regions called? They're called telomeres. Telo means tail towards the end. Telomeres they are called. Okay, right. So just want to review how the chromosome structure is so that you can understand the mechanism related to that. Centromere, chromatids, telomeres. Okay. Right. So what are chromosomal abnormalities to begin with? Chromosomal aberrations, these are also known as chromosomal abnormalities. Okay, right. So as I was telling you, these are abnormalities either in the chromosome structure or chromosome number. Okay, for your running note, simply write down chromosomal aberrations mean chromosomal abnormalities. Aberrations mean chromosomal abnormalities. These refer to either abnormalities in chromosome structure or chromosome number. Chromosome structure or chromosome number. Okay. So one structural abnormalities. So the problem or the difference in the structure of the chromosome. So chromosome structure gets affected here. I'll explain this later how. I think once we finish numerical abnormalities, I'll talk to you about this. Deletion. A portion of the chromosome is deleted. A portion of the chromosome is removed. This is called deletion. We'll discuss this. Just write like a flow chart. Make a note of this flow chart. Okay, write like a flow chart. So structural abnormalities, out of chromosome abnormalities, we have two types, structural and numerical abnormalities. Structural abnormalities refer to the changes in the structure of chromosome. Changes in the structure of chromosome. What changes can happen? Maybe a portion of the chromosome is deleted or removed. This is called deletion. Would that would that affect the chromosome structure? If you remove a portion of the chromosome, okay, it will lose the length of it, correct? And also sequence present in it. That is called deletion. Next one is duplication. You can easily guess what is duplication. I think in the last class we were briefly talking about duplication aspect as well. Copy variant, chromosomal copy variation, polymorphism, genetic polymorphism we talked about. Duplication means what? Adding additional information, duplication of the existing information, existing sequence. So instead of deletion here, extra chromosomal portion is added. That is called duplication. Addition of extra sequences. Okay, we'll discuss this with examples where possible and diagrams. I'll show you how they work. Deletion, duplication. Third is inversion. Just get familiar with the terms here. Deletion means deleting sequence. Duplication means adding sequence. It's like addition basically. Inversion means what? Existing sequence gets inverted. So you change the orientation of the sequence. It's like the CAT cat. It's supposed to be. Instead, it became TAC. So CAT cat, you flipped it. It becomes TAC. So meaning of it changes. That is called inversion, inverted. Okay, changing the orientation of the sequence. Next, translocation. Can someone guess what does translocation mean? The thing with biology is if you understand the terms, you can imagine it better. You can write it better. What does it mean by translocation?
Placement of a
Changing
sequence
the
is
location.
changed.
Changing the location. Trans means moving away from that location, shifting the location. So far, if you really realize deletion, duplication, inversion can happen within one chromosome. You remove a chromosomal portion, you add a chromosomal portion, you invert the chromosomal portion. But for translocation, we need multiple chromosomes. So you exchange the chromosomal information. How do we call that exchange of chromosomes or exchange of DNA sequences between homologous chromosomes is called as?
Recombination.
Exactly, recombination. You're switching the information between or sequences between homologous chromosomes. Translocation is not that. Translocation is where you can switch randomly with any chromosome, changing the information. Okay, you will understand it when I discuss about it. For now, just be familiar that these are called structural abnormalities. Only one example is given in the syllabus for this, that is for deletion. We have Cried-U-Chart syndrome that we'll not talk about. I'll explain that to you. Okay, I want you to be aware about this abnormality. This is about structural abnormalities. Next, we'll talk about numerical abnormalities. Numerical abnormalities. So numerical means number, changes in number of chromosomes. Okay, I'll introduce few terms here. First, write down euploidy. Write down euploidy. E-U-P-L-O-I-D-Y. E-U-P-L-O-I-D-Y. Euploidy, it is called. Euploidy. You see the spelling of here, ploidy, aneuploidy, polyploidy. We have two conditions. I'll explain that. Ploidy. Ploidy refers to number of chromosomes or number of chromosome sets. Ploidy. So ploidy. Okay. So euploidy that I told you. U means what? We talked about this in eukaryotes. U means?
Single.
U means true. Original in a way. Original. True. Okay. Euploidy refers to actual number of chromosomes. Actual number of chromosomes. Actual number of chromosomes. What is the euploid number in humans? So euploid is actual number of chromosomes. That is 22 autosomes, 22 pairs of autosomes plus one pair of allosome. 22 pairs of autosomes, one pair of allosome. Altogether 23 pairs of chromosomes. That is 46 chromosomes. That is a euploidy. That is the actual number of our chromosomes in each cell. 46 chromosomes. 23 pairs are. So here I want you to be aware about autosomes, allosomes, because in the syllabus they have given abnormalities related to this specific type of chromosomes. So 22 autosomal pairs and one pair of allosome. Together 23 pairs are 46 chromosomes. Correct? Autosomes are those chromosomes that supposed to be, that we perceive to be the chromosomes containing genetic information related to the somatic characteristics, bodily characteristics. And sex chromosomes were thought to be just meant for reproductive characteristics, but that's not true anymore. So we have reproductive kind of behavior controlled by autosomes as well as sex chromosomes. And some somatic characteristics are controlled by sex chromosomes. Like X chromosome is very important for our growth, for our bone density, for growth. So traditionally we call them sex chromosomes and autosomes, but they have overlapping functions. Okay. So now let's learn about these two terms, aneuploidy, polyploidy. Aneuploidy, polyploidy. So what is aneuploidy and what is polyploidy? First, I'll start with polyploidy. Polyploidy means what? Change in number of chromosomal sets. Polyploidy refers to change in number of chromosomal sets. Like for example, how many, we just talked about we have 23 pairs of chromosomes. What if you have one additional pair of chromosome? So what if we have not 46 but 69, or not 46 but double the amount of chromosomes, 92 chromosomes? So here in this abnormality, number of chromosome sets will increase by twofold, by threefold, by fourfold. So instead of 46, it may be 92, or it may be 69 if you have third pair. So normally the number of chromosome sets are referred by n, small n. We use small n to denote it. So remember we talked about diploid. Humans are diploid. We have 2n number of chromosomes, 2 times n equal to 23 in humans. Correct? Chromosome number is single set is equal to 23 in humans. 2 into 23, that is why we have 46 chromosomes. So instead of 2n, you may have 3n, 4n, 6n. That is called poly. Poly means many, ploidy, many sets of chromosomes. Many sets of chromosomes. So instead of 2n that is equal to 46, you may have 3n, there may be 4n, there may be 6n, like that. Have you heard about any humans with this number of chromosomes? Do humans exist with multiple sets of chromosomes, more than 2n? Have you heard about humans with 4n chromosomes, 6n, 8n? Is it possible? If you have not heard about, do you think humans exist like that? Instead of 46, have you heard about people with 92 chromosomes in their cells? And we're talking about after cell division, not during cell division process, okay? Even during cell division process, we'll never have 92, but we'll have sister chromatids. Any idea? Any guesses?
Transgender cases. Transgender cases.
Transgender cases, okay. Transgender, we'll talk about transgender story as well. Transgenders don't have extra sets of chromosomes. So humans cannot tolerate multiple sets of chromosomes. We can tolerate one chromosome extra or one chromosome less, but we cannot tolerate to have multiple sets of chromosomes. Even having one extra chromosome or one less chromosome creates enough problem in us. How can we tolerate to have entire set of chromosomes extra? No, it doesn't happen. In plants, you might have seen some hybrid varieties which actually have a bigger in size. Have you seen pumpkins, huge pumpkins, sometimes as big as a room? There are competitions that happen to grow such pumpkins. Those type of pumpkins have multiple sets of chromosomes. That is called polyploidy. They have 8n, 16n. So 16 times the chromosome number. There is some correlation between number of increase in number of sets of chromosomes and the size of such a plant, for example, such a plant product like pumpkin, brinjal. Some plants, actually we use polyploidy to grow them in large sizes, in large quantities. But humans cannot tolerate. So we don't have human examples for polyploidy. But this is part of the basic idea, so I wanted to introduce. So we're not going to learn about polyploidy. Just be familiar what is polyploidy is about. Okay?
Is it observed naturally, sir, in plants or?
Good question. Is it observed naturally in plants? I believe so. I don't remember exactly whether, but yes, as far as my understanding goes, you can even see it naturally. Let me cross-check that. But otherwise, yes, humans actually introduce those changes as well. There are a few chemicals which don't allow the separation of chromosomes and allow them to multiply. Okay, this is definitely done synthetically. And I do believe that there are natural existence of that, but I have to cross-check once. So let me make a note of that. And I'll tell you that in the next class. Okay. Okay. Right. So, but we don't have human examples because humans cannot tolerate this. So what is common in humans is the aneuploidy. Aneuploidy. What is aneuploidy? Can you guess? Based on what we discussed about ploidy means number of sets. Poly means many sets of chromosomes. Euploidy means true or original number of chromosome sets. What is aneuploidy?
Reduction in number of sets.
Reduction number of sets. Okay, it is not reduction number of sets, but it is reduction in number of chromosomes. Because sets we cannot tolerate. You cannot have 23 chromosomes unless during the meiosis, during the formation of gametes. So, but chromosomes can reduce individually by one, usually by one. Change in individual chromosome number. Change in individual chromosome number is called aneuploidy. Excuse me. So aneuploidy refers to change in individual chromosome numbers. So most of the examples we'll discuss in this chapter belong to aneuploidy. They belong to aneuploidy. Do you understand the difference between polyploidy and aneuploidy? In polyploidy, we have chromosome sets being either 2n to 3n to 4n, 6n, 8n, so on. Whole sets increase, but we cannot tolerate. We may see examples in plants. Aneuploidy is change in individual chromosome number. So 46 chromosomes may become 45, or 46 may become 47. So maybe one extra chromosome or one less chromosome. We can't tolerate much more than this. All the examples you see in the literature, one example for reduction in chromosome number, that is 45. Other examples are additional chromosomes, one extra chromosome. So having 47 chromosomes. So do you see it here? What is changing? Only one number, either one chromosome got deducted or one chromosome got added. Such individual change in chromosome numbers is called aneuploidy.
That means one of the chromosome is independent, like not in pair.
It is in pair. It is actually because of an abnormality that we'll talk about, because of a problem in cell division, this happens. So cell receives more than 2 chromosomes, it receives third chromosome as well. So there will be 3 chromosomes in one cell with respect to that particular. Okay, I'll show the images for you to understand on that. So all the examples many of the examples, leaving out one, are all examples for aneuploidy. Either the things given in 9.4b, things given in 9.4c, as I was already telling you. Okay, leaving out Craig-Duchat syndrome, they are aneuploidy. So they're the examples of aneuploidy. You need to know this because in the exam, if they ask you to discuss the examples of aneuploidy, you need to know what to write. So terminology is very, very important. All this is given in B and C belongs to aneuploidy except for one. Okay, and B and C, as I was telling you, you can write aneuploidy, put 2 arrow marks to aneuploidy. So aneuploidy, or change in number of chromosomes, either may happen in autosomal chromosomes or sex chromosomes or allosomes. So put an arrow mark to aneuploidy, 2 arrow marks, 1 arrow mark should, and you need to talk about autosomal abnormalities, autosomal aneuploidy or aneuploidy in autosomes. So chromosomal variation, number variation in autosomes. Another arrow should refer to 6 chromosomal abnormalities or aneuploidy in allosomes or sex chromosomes. Aneuploidy in allosomes are sex chromosomes. Okay, we'll learn about enough examples from B and C, but this is a brief overview about chromosomal abnormalities, types of them. Euploidy, aneuploidy, polyploidy we talked about as part of numerical abnormalities. In structural abnormalities, we talked about deletion, duplication, inversion, and translocation. Okay, this is basic overview about types of chromosomal abnormalities. Now, what causes this number changes? We'll first focus on numerical abnormalities. What causes changes in chromosomal number? So here we are learning the mechanism. So why do we get 1 chromosome extra or 1 chromosome less? There must be something going wrong. And this has to happen somewhere during the cell division process. This is where chromosomes are segregated, right? If the segregation process doesn't happen as it's supposed to, it may leave extra chromosome to one cell and 1 less chromosome to the other cell. Correct? Is that possible? It's like this. You have 4 chocolates and you have, let's say you have 2 brothers or sisters. One, you can give 2 to one sister and 2 to another sister. But what you actually decided to give 3 to one sister, 1 to another sister. Isn't it abnormal or not? Okay, it is like that. So instead of 2 chromosomes separating, sometimes what happens, like we have sister chromatids, sister chromatids supposed to separate like this. But instead what happens? Both goes to one side, leaving nothing on the other side. Okay, I'll explain that with this image. So the process behind this aneuploidy is called non-disjunction. I'll explain this concept. This is a mechanism behind chromosomal numerical abnormalities, or more specifically behind aneuploidy. Excuse me. So non-disjunction. Disjunction means what? Dis means separate, junction. Something that is together, separating it, moving it away is disjunction. Junction means together, disjunction means moving it away. Non-disjunction means not moving it away, not separating it. So it's a bit redundant. You can call it junction, correct? It's together. But how do you know it is not just the process before cell division, or it is because something went wrong during the cell division? Although disjunction is supposed to happen, but it has not taken place. It's called non-disjunction, non-separation of chromosomes. Okay, which is an error in cell division. Which is an error in cell division. Can happen in mitosis or meiosis both. But as I was telling you, I think what is more significant is in the meiosis. An error in cell division in which members of a pair of chromosomes fail to separate. So it's an error in cell division where member of pairs of chromosomes don't separate or fail to separate. This is non-disjunction. So during cell division, where it's supposed to separate individually into gametes, that doesn't happen. So we are interested in talking about gamete formation here. So remember meiosis, what happens? How many cells are formed in meiosis? How many daughter cells are formed?
Haploids.
How many haploids are formed from one cell?
4.
4 haploids.
4 daughter cells are formed, right? 4 daughter cells are formed from one cell. Okay, they should have one copy of each chromosome. Okay, it should have 23 chromosomes in each cell. So what happens here? This image explains the mechanism. So this is a diploid cell, correct? This is a diploid cell. Diploid cell has 2n, 2 times the chromosome. This is one chromosome, this is another copy of the. This is another chromosome, this is another copy of the chromosome. From this, after meiosis, we should get a cell like this with n. So haploid gametes we need to get, correct? Haploid means one copy, not 2 copies, no sister chromatids. Only one chromatid and one chromatid. The 2 chromosomes all together in this theoretical example. So there should be one chromosome of one second chromosome, or A and B. We should have a single chromatid of each chromosome. This should be the result of meiosis. So N should look like this. This is normal. This is what we normally need to have. But sometimes what happens because of the non-disjunction. So as you see here in the centromere, they both are like this. They're supposed to separate from each other. Chromosomes are supposed to go like this, go away like this, but they don't. It is too stuck, too much, too much glued in a way. Okay, they're too stuck together. So what happens, instead of both of these should separate into 2 gametic cells, instead both of these goes into one cell. So what happens because of that, you get one extra chromosome. So we have 2 chromosomes. This is normal. There's the 3rd chromosome in the cell. There's supposed to be only 2. It's like in human cases, supposed to be 23. But you got 24 chromosomes in one cell, one extra. So in this theoretical example, there's supposed to be 2, but there's an additional chromosome. That is why it's called n plus 1. N is the actual number, haploid number of the chromosomes, plus 1 is one additional chromosome. So n plus 1. This diagram is very, very important. And you keep getting these questions. If you explain it this with the diagram, this will be really, really useful. This will be really great. Okay, you will have this diagram explaining how non-disjunction takes place. Starting with a diploid cell, we're supposed to get gametes like this, but another possibility is where you got one extra chromosome because of the non-disjunction, non-separation between a chromosome pair. So this results in n+1. On the other hand, the other cell might have not received this chromosome, right? Because it didn't get separated, it went to one side. What about the other cell? This chromosome didn't go into other cell. So you lost one chromosome. The cell lost one chromosome. So this became n-1. Instead of getting two chromosomes like this, it only got one chromosome. It only got this small one. This large one is gone. This is another condition. So either adding one extra chromosome or removing one chromosome happens during the non-disjunction process. One gamete receives two chromosomes, another gamete receives one less chromosome, one chromosome less than what it's supposed to have. So one extra is called n+1, one less is called n-1. Now, so when you get these type of gametes formed because of the fertilization, when fertilization takes place, when mating with the normal gamete takes place, this is normal gamete. When N mates with N, what will be the result? N into N. 2N. 2N, right? So we'll get something like this. These two chromosomes got multiplied and we have sets of them, copies of them in the diploid state. So when two chromosomes like this fuse, after replication, you will find homologous chromosomes like this. This is a 2N condition, diploid condition. So N mates with N, normal gamete with another gamete, would result in 2N product, 2N zygote. Excuse me. Instead, if you have n+1 mates with N, something like this. Imagine this is a gamete. This n+1 mates with N. What will be the result? Something looks like this, 2N condition plus one more chromosome with the sister chromatic attached to it. So this is a 2N+1 condition. N+1 mates with N resulting in 2N+1. This is called trisomy. This is called trisomy. Be familiar with these terms. Trisomy. Trisomy. One example we'll discuss here is 21 trisomy. 21st chromosome received 3 chromosomes instead of a pair of chromosomes. 3. This is called 21 trisomy. Third chromosome. Tri means 3. Trisomy. Third chromosome is present in this cell. This is called, for example, this is the case with Down syndrome. Down syndrome. I'll show the images of them that you might have seen people with Down syndrome around you. Very rare, but you do see people. This is called trisomy. 21 trisomy if it is Down syndrome. Otherwise, simply we call it trisomy, one extra chromosome. Instead of 2, there are 3 with respect to that chromosome. Similarly, in the case with n-1, if n-1 mates with normal N, normal gamete, what will be the result? This chromosome will be missing, correct? What we have is these two. 2N state for these two. Plus we have this additional, we have this chromosome, one chromosome. So n-1, because this one chromosome and this chromosome came from the N. These two came from N. This came from this. So totally pair of this, pair of this, pair of this. Totally 3 chromosomes you will have. So to diploid number, it is 2N-1 because it's supposed to be like this. It's supposed to be 2N. We're supposed to have 4 chromosomes like this, but we only have 2. So we only have 3. So it's 2N-1 is this. This is called monosomy. Instead of 2, there is only 1. So mono. Instead of 2, there were 3. That is trisomy. So this is how chromosomal abnormalities happen. Numerical abnormalities happen due to non-disjunction process. This is a well-known mechanism. Any questions on this? Did you understand this? Is there anything that you could not follow here? I'll review this, see if you followed this mechanism. What are we learning here? How does chromosomal abnormalities take place? And here we're talking about numerical abnormality, which is aneuploidy. Okay, I'll ask question, see if you can answer it while I'm revising this. Okay, so this becomes, it will give you a little confidence if you're able to answer them. So what is the normal state of chromosomal number in our cells? Are we diploids or haploids?
Diploid.
We are diploids.
Diploid.
Gametes are diploids. So we are diploid organisms. And when diploid organisms, okay, undergo meiosis, it would result in haploids or diploids. End of meiosis. What would
Haploid.
we receive? Haploid cells. How many of them we receive?
4.
We talked about it already. 4 haploid
4
cells.
haploid cells.
Single cell like this. Okay. But we're supposed to have haploid number like this if there are only 2 chromosomes. But what, for some reason, you got 1 extra chromosome. What can result in an extra chromosome? What process can result in n+1 condition? What process are we learning about?
Aneuploidy.
Okay, aneuploidy. What are we learning here in this slide?
So non-disjunction.
Exactly, non-disjunction. Disjunction means separation. Non-disjunction means no separation. So if 1 chromosome didn't get separated, you will have that extra chromosome sitting in the cell, sitting in the gamete. So resulting in n+1 condition, n+1. So instead of these 2 chromosomes, now we have 1 additional chromosome. Similarly, on the other side of the cell, what would you notice? Non-disjunction would result in 1 less chromosome in 1 gamete, because this chromosome has gone to this gamete, leaving only 1 chromosome in this gamete. So this is n-1 condition. N is the haploid number -1, 23 minus 1, 22 condition in humans. N+1, 23 plus 1, 24 chromosomes. This is the gamete stage, gametic stage, when gametes are formed. But what will be the product if these gametes participates in mating or fertilization process? The result will be this. Normal gamete, when meets with normal gamete, you will get n into n or n+n, 2n you will get. Whereas n+1 meets with n, it will result in 2n plus
2n+1.
n+1 mated with n. So 2n plus 1. Those 2, n will bring 23 chromosomes. Another n will bring another 23 chromosomes, 46. Plus 1, that extra chromosome. Altogether 47 chromosomes. That condition is called trisomy, can be mentioned as 47 chromosomes. We normally write the chromosome that is extra. I'll show you that in the coming slides. 47, let's say Down syndrome. Or let's say we'll talk about Klinefelter syndrome. Okay, we'll write 21 XXY composition. Okay, I'll explain that to you later. That is called trisomic condition. Trisomy. Next, monosomy. So n-1 fertilizes with gamete with normal number of chromosomes. Result is n-1 into n. What will be the result?
2n minus 1.
2n-1. You see it here in the image. Are you able to follow this? You should be able to write it. The diagram, you should be able to draw it yourself. So what happened here? N-1 joined n. So basically you have 23 coming from the normal gamete. 23. From here you only have 22. So meaning 46 minus 1. So 2n minus 1. This is
45.
monosomic condition. Yes. So 2n is 2 into 23 minus 1. That is 45, right? 46 minus 1, 45. This is monosomic condition. So would you be able to draw this diagram and explain how chromosomal abnormalities, numerical abnormalities take place? Okay, this is about the non-disjunction process that I want you to be familiar about, and write this when question on numerical abnormalities is being asked. Okay, can we move on? This I have taken from a textbook which talks about incidence of chromosomal abnormalities in newborn infants. Newly born infants have what number, what percentage of chromosomal abnormalities? This is the incidence rate, and you see correlation with the maternal age. In this chart, you notice that here they talk about maternal age of the mother, 20 years, 30 years, 35, 40, 45 years. What do you notice here? Incidence of Down syndrome. I just told you, what is Down syndrome? One extra 21st chromosome is present in some children. 21 trisomy. Third chromosome that we talked about here in 2n plus 1. This extra chromosome happens to be the chromosome of 21, 21st chromosome. That is itself called Down syndrome. So read this and tell me what are you noticing here. Age 20 years, incidence of Down syndrome, 1 in 1667. One child in 1667 children at age 20 shows Down syndrome.
As mother's age increases, the child she gives birth to has a higher chance of having a Down syndrome.
Exactly. So there is higher chance of getting Down syndrome. So with increase in maternal age seems to increase the incidence of Down syndrome like abnormalities. So one factor here is mother's age, maternal age. 1 in 1667. Next, what happened at age 30, if you consider, there's a higher chance of having a Down syndrome baby. 1 in 952. 1 out of 952. 1 less than 1,000. Before it was 1 more than 1,000, more than 1,500. So 1 in 952. Higher probability.
So incident like Down syndrome and trisomy is like same.
Downs is an example of trisomy. Trisomies can be any of these. Patau's, Edwards, Klinefelters, all these are trisomic examples. Down is one trisomic condition. Trisomy means having 3 chromosomes. It could be from the literature, from observation, we know 3 chromosomes are possible with X chromosome, with 21st chromosome, with chromosome 18, with chromosome 13, like that. Few chromosomes are possible. So Downs is one such condition, one of those examples. Okay, trisomy is a general term. Downs is 21 trisomy condition.
Okay,
So 1 in 952, 1 in 385, 1 in 106. At the age of 45, there is a chance of having one child out of 30 with Down syndrome. So with increase in maternal age, chances of Down syndrome is also increasing. More or less, this is the case with any other chromosomal abnormality. Slightly less variation can be seen here, but even there, starting from at the age of 20, maternal mother at age of 20, if you compare, they will show 1 out of 526 with Down syndrome, and this probability will increase as the age increases. So this is true with many chromosomal abnormalities as well, although at the same rate, but this is also true.
So here, father's is of no importance.
Father's age, there are few aspects of father's age can matter, but not the. So as you notice, because one aspect that you notice is the environment in which cells are divided. So mother's age seems to play a bigger role or have a bigger impact in this case. Okay. Next, we'll talk about the overview about B and C. Here I'll briefly tell you or introduce you the diseases that are given in B and C. Then we'll discuss each of them individually in detail, because they're asking short notes on these diseases in the exam. So examples of chromosomal abnormalities, and most of these are given in the syllabus. Okay, first look at the bottom one, sex chromosomal abnormalities. So 1.4b, sex chromosomal abnormalities. We have to learn in detail about them. So now you should be able to recognize. Look at the number of chromosomes. 47, 45, 47, 47. 47, trisomy or monosomy? 47 condition. Is it trisomy or monosomy?
Trisomy.
Trisomy. 47, 46 plus 1, 2n plus 1. 45.
Monosomy.
Monosomy, exactly. But here, what is extra? So these are all 6 chromosomal. You notice here we are referring to only 6 chromosomes. In this case, what is extra? You see XXY, 47 comma XXY, which is also known as Klinefelter syndrome. Is also known as Klinefelter syndrome. What is extra here?
X chromosome.
X chromosome is extra. Why are you saying X is extra here?
From mother's side.
So here it is a male, XY is supposed to be, and then it got one extra chromosome. One extra X chromosome. Okay? Supposed to be XY, but it got extra X. Okay? So this is Klinefelter syndrome. What is Turner's? Is a monosomic condition. 45X. Are there chromosomes extra or less here? Do you see more chromosomes or less chromosomes compared to normal number of chromosomes in our cells?
Less chromosome.
Less, one chromosome less. We're supposed to have 46. There's only 45. Okay. So this is called Turner syndrome. What is less here? What chromosome is less?
Why?
Y. So this is X chromosome missing here. Supposed to be a female with XX, but one chromosome is less. So X, or in some combinations, maybe Y is missing, depending on what is other gamete, right? But normally what happens, Y cannot survive by itself. The reason why I'm telling you that X is missing here is Y by itself cannot survive. Gamete with Y cannot survive. Otherwise, we'll also have Y naught, Y with no chromosome, but that condition doesn't exist. So Y by itself cannot exist, so it is supposed to be X that is less. Okay, this is called Turner syndrome, named after those scientists who observed or identified these conditions. Klinefelter is the name of scientist, Turner is the name of scientist, Jacob is the name of the scientist. Okay, right. Sometimes we also see 3X chromosomes. One extra X chromosome along with 2X chromosomes. Triple X it is called, or 3X chromosomes. 47XXX we write. Okay, we'll discuss the details, specifics of these phenotypes. Don't worry now. I'll explain them one after the other. Okay, so this is sex chromosomal abnormalities. Klinefelter's, Turner's, Jacob syndrome, and triple X. 47XXY, 45XO or X0 we call, 47XYY, 2Y chromosomes, and 47XXX. Okay.
Is it helpful to remember the phenotype
Yes.
as well?
This one, a phenotype, yes. We are going to discuss about that, but I will show the image and discuss that with you.
Okay.
Instead of by hearting from the table here, I show the image for that and show it. You have to write the phenotypes, but I want to show it separately in a different slide. Here, we're just providing an overview about types of them, names of them. Okay. Similarly, in 1.C, we'll talk about, see, these are all sex chromosomal abnormalities. Do you notice? Can you recognize them? Here we're talking about addition of X chromosomes or Y chromosomes here. XXY. Because normal condition is for females XX. Right? So we have two possibilities here, two deviations from here. One, from XX, which is normal female, 46 comma XX we write. 46 comma XX we write. Here we have 45 comma X, meaning one chromosome is reduced. X0 we call sometimes, X0. 45 X0. Or there is one additional chromosome, 47XXX. That is triple X or super females it is called. Super females with one extra X chromosome. Similarly, deviation from normal males, 46XY is a normal male. Is it possible to have 45Y0? Is that condition possible? We're talking about deviation from normal males. Normal male is 47XY. Sorry, 46XY. Is 45Y0, is it possible, or not Y? You have to leave out the X chromosome. Not Y or 0Y or Y0. Is it possible?
No, sir.
Why? Why is that?
So because females have XX and at least one X would be coming.
So 1X is needed. It is essential to have 1X. Both females and males contain X chromosome. Females have 2 and males have 1X.
1X.
So X is essential. Without X, we cannot survive. Okay? Right. So Y0 is not possible, but 47XYY is possible. This is also called super males. This is called a super male. So it's not a superman, it's a super male. Okay? With 47XYY, males don't become superman, they become super males. Okay? So these are sex chromosomal abnormalities. Develop this comparative idea and write incrementally what is normal for females. If one chromosome is less, one chromosome is extra. How are they called? You have to by heart these names as well. 45X0 is Turner's. 47XXY is Klinefelter's. Different from Jacob syndrome. This goes in combination. Super female, super males. Super means one extra. Female means extra X. Male means extra Y. XYY, XX. So this genotype you have to write. This genotype is also very important. So I'll give you a structure as well for the answer. If you get a question like this, for Klinefelter's and those syndrome, what needs to go into it? Okay, these are sex chromosomal abnormalities. Briefly, what are the examples for autosomal abnormalities? Here we'll only learn about one such case where 45 chromosomes are there. Rest of them are trisomies. So I was telling you already 21 trisomy or trisomy 21 is also known as what syndrome? Down syndrome. Trisomy 21 is also known as Down syndrome. Okay. Similarly, not only trisomy 21, trisomy 13, trisomy 18 is same. So trisomy 13, 3 chromosomes of 13th chromosome is seen in some people. It is known as Patout syndrome, again with the name of the scientist. And trisomy 18, 18 chromosomes found in an individual is suffering from Edwards syndrome. Is suffering from Edwards syndrome.
Sir, here 18, 21, 13 are the number of trisomies.
The number of the chromosome. Which chromosome? So autosomes, we have 22 pairs of autosomes, right? We number them 1 to 22. When we say trisomy 13, the 13th chromosome in that has 3 copies. Instead of 2, 13th chromosome has 3 copies. When we say trisomy 18, 18th chromosome has 3 copies. These are trisomic conditions of all of them. 47 chromosomes are there here too. Okay, but instead of writing 47 XXY for autosomes, what do we write? We say we give that specific number where abnormality is there. So chromosome 13 has trisomic condition. So 13th chromosome has additional copy of chromosome. Similarly, trisomy 18, trisomy 20. And they have names. 13 is Patau's, 18 is Edwards, 21 is Down syndrome. Okay, right. From the list, tell me, you also have incidence rate here. Can you tell me which of all these diseases, which of all these chromosome abnormalities are common? Which of these are common? Relatively. Which of these are more common out of all these things?
Klinefelter and Down syndrome.
Klinefelter and Down syndrome, these two are the most common of all the chromosomal abnormalities that are given here. Why? Because Klinefelter can happen 1 in 500 males. 1 out of 500 males may be Klinefelter. This is the incidence rate. You may see slight variation in number. If instead of 500 in some textbooks, they say 600, 700, don't worry. That's not too much of a difference. Instead of saying 1 in 500, if they say 1 in 5,000, something might be wrong. But these are all standard books of genetics. Okay, this is taken from Lewis, Human Genetics, very standard human genetics book. Okay, so 1 in 500. So Down syndrome is one common. So, sorry, Klinefelter and Down syndrome are 2 common diseases, chromosomal abnormalities. Here it happens 1 in 800, this happens 1 in 500. What is the least common of all these things?
Patau syndrome.
Patau syndrome. After that,
Edwards syndrome.
Edward. These are very complex and dangerous diseases. Not many children would survive. Even these children with trisomy 13, 18 has a difficulty to survive. So they're dangerous. So some people may die in the womb itself. They might not even survive. They may not even born. Stillbirths may be seen in some people. So they're dangerous. They have more severe complications. So their incidence rate is less. 1 case in 15,000 cases. Here 1 case in 7,500 cases. And rest of them comes in between. The more common are Down syndrome and Klinefelters. Then comes the XYY and so super females and super males, 1 in 1,000 case. And then comes the Turner syndrome, 1 in 2,000 females. Then comes the trisomy 18, 1 in 7,500. Then trisomy 13, 1 in 15. Roughly, because this incidence number is very, very important for you to write in the exam. The reason why I'm actually giving this comparative analysis because it will become easy for you to remember. Down syndrome. So we know that it is less than 1 in 1,000 cases. So approximately 1 in 800. Klinefelter 1 in 500. What comes next? 1 in 1,000 is super females and super males. What comes after that? 1 in 2,000 Turners. Then comes the Edwards, 1 in 7,500. Then comes the cutoffs, 1 in 15,000. So roughly remember this. Okay. If you go wrong by few hundreds, it's all right. But if you go wrong by few times, instead of 1,000, 2,000, instead of 500, 5,000, that'll be wrong. Okay. Right. But repeatedly, when you read this, you'll be able to remember this. Right. Now let's get into the details. Is this clear to you? 6 chromosomal abnormalities examples, autosomal abnormalities examples. Now let's look into the individual chromosomes, starting with the sex chromosome abnormalities. So under numerical aberrations, so this we are here now, we move to 9.4b. Leaving out structural aberrations, we discussed about what are chromosomes, chromosomal aberrations. Okay, we are now talking about 9.4b, sex chromosomal aberrations, and specifically numerical aberrations. So first we'll start with Klinefelter syndrome. Klinefelter syndrome. So first two you already know. So you have to write first the genotype of this. What is the genotype? We already discussed about genotype. Klinefelter's is XXY, 47XXY. Either you write 47, XXY. Normally the textbooks they like 47, XXY, they write. So write like that instead of hyphen. 47, XXY. So trisomy at related to the sex chromosome, X chromosome. And why is it called Klinefelter's? Because Harry Klinefelter from Massachusetts General Hospital first identified this. That is why on his name, it is called a Klinefelter syndrome. Next, how common is this? What is the incidence rate? 1 in 500 cases. 1 in 500 males show Klinefelter's. So one of the most common chromosomal abnormalities. Okay, so something that you can easily derive and remember, are they males and are females? XXY is a male or a female?
Male.
What if you actually have, instead of XXY, you have XXXXXY? Would that be a male or a female?
Male.
5 X chromosomes, 1 Y.
Male.
More towards female.
More towards female.
It will be a
But
male.
it's a male.
It is a male, but they have that female, or probably female with some male characteristics.
Gender wise, even if you have 1 Y chromosome, it is a male. Why? Because Y is the sex determining chromosome. Y is the gender determining chromosome. X doesn't have a role in this. Y chromosome has a role. I'll tell you briefly the concept related to this, but we'll discuss more about this in unit 10, human growth and development. So normally when we are born, okay, when zygote is formed, when embryo is beginning to happen, okay, we have what is called bipotential gonads, meaning both females and males have a very similar start. They can become either a female or a male. By default, all of us are females. In mother's womb, when the conception happens, all of us are by default are females. So we start life together in the same step, same way we're starting to develop, becoming embryo. Signal to define the gender comes in about 6 weeks. About 6 weeks of time, Y chromosome, there is a region on the Y chromosome which is called sex determining region on the Y chromosome. Sex determining region on the Y chromosome. So from this a protein comes and that protein acts like a signal. So only in presence of Y chromosome where this protein gets released or expressed, that zygote will give rise to a male. In absence of this signal, by default, we are all females to begin with our life. Okay, so signal is coming from Y chromosome. So Y is the gender determining chromosome. That is why in absence of Y, it will be a female. And it doesn't matter how many X chromosomes are there. 5, for example, even if there is one Y chromosome, that will become a male. That will become a male. So theoretically, they normally talk about this. 49, for example, XX, XX, Y. Even that is a male. For theoretical. Because your importance is Y chromosome has a gender determining ability. That is why I was asking you the difference between XXY and XXXXXY. Okay, is it clear? So Y is a gender determining factor, and when is gender determined during our life? Second month, 1.5 months, 6 weeks is when gender is determined. What determines it? A gene on Y chromosome. Okay, right. So that is why by looking at this, if all X chromosomes write genetically females, if one Y chromosome is present, write genetically male. Okay, right. This is easy to write. Incidence, name, this. Next, features. So people with this Klinefelter syndrome, what features do you notice in this? For these type of things, you have to prepare to draw some rough diagrams. For this and also for fossils, unit 1.6, where we draw the structure, skeletal structures. This is where you have to practice some diagrams. Here, only highlight few problematic things. You don't have to remember all of them. I'll help you to organize this information. Okay, right. So XXY. Look at few things here. Look at first any growth abnormalities. See if there are any growth abnormalities, which are visual, which are obvious to us. And think about any behavioral abnormalities. Okay. Growth abnormalities when they're born, and any behavioral abnormalities, any mental problems, things like that. Here, features. So it's supposed to be XX, supposed to be XY. Extra X chromosome results in more height. So people with XXY chromosome tend to be really tall. You see this person is usually tall. You can even find on Google some images where if you have a normal male versus male Klinefelter's male, even super male, they tend to be tall. And especially here, where is this height coming from, this additional height coming from? Not because of the trunk, because they have long limbs. They have long legs and long arms. So they are taller with longer arms and legs. As depicted in the image here, they have long legs and long arms. But trunk is more or less on the average size of normal males. Okay, very, very important aspect. This is the first simple aspect is height, that they tend to be tall. Hypogonadism. The most important feature of them is hypo. Hypo means what? Hypo, hyper. Are you familiar with these terms? Hypo, hyper. Hypo means more or less.
Less.
Less. Hyper means more. Hyper. Hyper. I don't know if you have seen this supermarkets in Hyderabad. We have something called hypermarkets. Hyper, they're bigger. They're larger versions of supermarkets. Hyper. Hypo means less. Hypothyroidism, hyperthyroidism. Hyperthyroidism means producing more thyroid hormones. Hypo means producing less thyroid hormones. So this is all factual, okay? It all depends on how do you connect this information and how much do you remember. Otherwise, this is factual and these questions usually come in the exam. Okay, so hypo means less. Gonadism means gonads refers to the glands in males which release testosterone hormone. So testis. So testis is what releases testosterone in males. Sperms as well as testosterone hormone is released by testis in males. So male reproductive organs release hormone called testosterone. And testosterone is responsible for male characteristics. male characteristics and less of testosterone. Because of extra X chromosome, testosterone production is reduced. Then what happens? So what will be the impact of hypogonadism? Reduced male characteristics. Less signal. Hormone is a signaling molecule. Hormone defines the characteristics, especially in the reproduction process. So when less testosterone is produced, lesser the output from that. So reduced secondary sexual characteristics are seen. Male-like characteristics are reduced. So for example, frontal baldness. They will normally have less hair here in the front. Frontal baldness. And poor beard. They don't have good beard growth. Poor beard, which is a secondary sexual characteristics in male. Less beard, less hair in the front especially. And you see in the image, tendency to lose chest hair. Hair on the chest is also less. Hair on the chest is also less. So reduced male characteristics, first part of this. Hypogonadism, reduced secondary sexual characteristics. So frontal baldness is absent. Sorry, I think I got it. Frontal baldness is absent. Not reduced hair here, but they normally have the absence of this process. Normally, males tend to have lesser hair, here, but that baldness is absent. Okay, so poor beard, less beard. So reduced male characteristics, absence of frontal baldness and poor beard growth. Next, because of extra X chromosome, feminine characteristics are seen. So feminized physique, slightly feminized physique. Feminized physics, sorry. So you don't have to use these terms. Like gynecomastia, if you're comfortable, use it. Gynecologist, you know gynecologist, right? Okay, one who actually deals with female issues. Gynecologist. So if you can remember gynecomastia, otherwise, poor breast development, poor development of breast. Okay, or if you are seeing in the male characteristics, development of breast. So breast development, 30% of cases, they have feminine characteristics here, female-like characteristics here, reduced male characteristics plus female characteristics. So if you remember this theme, it won't be too difficult for you to write about reduced testosterone, poor beard growth, and add some feminine characteristics. Like breast development is seen in them in 30%. Not all of them should have same characteristics here. Overall, an average, nowadays we are hearing for COVID, right? These are all the possible symptoms. Not all of them will have all symptoms, okay? But 30% of cases have breast development. And slightly feminized appearance. As I was telling you, slightly feminized the physique. Overall shape of the body is more feminized. Okay, right. So there are more characteristics here that I have not talked about in addition to poor beard growth, small testis size, and female-like hair pattern, pubic hair pattern. So these are all some female characteristics and lack or reduction in male characteristics. Okay, then. So this is about the characteristics, physical appearance-wise. Next, infertility. If there is less testosterone, less sperm production, and infertility is common in them. So they cannot have children. Infertility is commonly seen in people with the Klinefelter syndrome. They tend to be infertile. Next, their IQ mentally. Next, we are going from physical components to the psychological and mental. So they seem to have reduced or slightly impaired IQ. Their intelligent quotient is also decreased, slightly reduced IQ. And they show issues in learning. They show learning disabilities, issues in learning. Their reading ability, spelling ability is actually compromised. So they're not quick learners. They slowly learn. The reading and spelling is difficult for them. Because of, and as you see it here, XXY, 6 chromosomes, but still affecting the body growth, affecting the performance of the brain. So this clearly shows, this abnormality shows the role of X chromosome, either it is extra or normal. So this is impact. Okay. So they have normally learning difficulties, learning disabilities. So if you don't remember all of these examples, even if you remember a few of them, that would be sufficient. Okay. Next, if you can add some additional ones, it's great. Like something like clinodactyly is where people have bent fingers. Fingers are not straight like this. They usually have bent fingers like this. This is called clinodactyly. Okay, you can omit this. If you cannot remember these terms, this is sufficient to write because you don't have space for writing all this. So you choose some of these: reduced male characteristics and some feminine characteristics, infertility, reduced IQ, learning disabilities, increased risk. So these people tend to have more bone related issues: osteoporosis, loss of bone density, osteoporosis, or more porous like bones, weak bones, because hormonal problems. Diabetes, they tend to have higher incidence of diabetes. Also, certain cancers are more common in them. If you can remember this, great. Otherwise, this first part is what you need to write most. These points. Okay. So you mention about incidence, you write about genotype of the disease like this and say whether they're genetically males or females. So this you can easily remember and try to remember the features based on the logic I explained you and what happens in them. Okay, but next you can talk about how do you diagnose them? How do you know that they have Klinefelters? Diagnosis, you put a subheading called diagnosis. How do you diagnose them? So we learn about some methods, genetic methods in unit 9.1. Okay, I'll bring this back again when we discuss about 9.1 as applications of those methods. But for now, I'll just briefly show it. Any idea? Anyone knows what is the answer to this? How do you diagnose if someone has Klinefelter syndrome? What are the possibilities? Vishal, can you answer this question?
No, sir, I can guess.
Yes, yes, please.
I think maybe one of these symptoms, if they manifest
Okay.
in a...
Clinical features. One way is clinical features, but problem is sometimes there's overlap of in some features. How do you know this is exactly the cause of the problem? Because some hormonal imbalances can also cause testosterone. Reduction in testosterone can happen in males sometimes. Okay, and sometimes estrogen quantity may be high. They're genetically normal, but they have higher amount of estrogen, or some of the foods that they're eating has estrogen present in them. Can also result in some of these abnormalities. How do you know whether it is a chromosome abnormality versus hormonal imbalance? Your point is right. I'm just adding the logic to it. So what's causing this problem here? So what's causing the problem?
extra chromosome.
Extra chromosome. Exactly. Is there a method to identify the extra chromosomes? Or is there a way to identify what chromosomes we have in our cells? Can we take cells and somehow open them and count the number of chromosomes? Is it possible?
Yeah, I think yes.
It is possible. Okay, this is called karyotyping. So this is something that we are going to learn in unit 9.1 as a method to study humans. Okay, so what are you seeing here? I don't get into details, but we are going to talk about this. What is this method? How are these chromosomes created? How do you count them? What are the advances? We are going to talk about this in unit 9.1. For now, there is a method called karyotyping. If you want in diagnosis, write down karyotyping. K-A-R-Y-O. Karyo. K-A-R-Y-O. Karyotyping. Karyotyping. I'm typing it here. So karyotyping is a method. So diagnostic method for chromosome and abnormalities is karyotyping. So what are you doing here? Briefly, I won't get into the details, but what you see here, this is chromosome 1. We need to have a pair of chromosome 1. Okay. They normally know the size, length of them, and they know some pattern they create. They stain them with some colors. Okay. And you have a pattern that matches here. Exactly same, thickness or darkness you see in the bands here. Based on that and based on the size, they would actually make them, group them. Like that, you see 1, 2, 3, 4, 5 here and up to 12 here, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Totally 22 pairs of autosomes, 1 pair of sex chromosome. So 22 pairs you have here, right? Do you see abnormality here? The 2 X chromosomes and 1 Y chromosome. What is the genotype here? So
XXY.
22 plus 23 in position of 23. Here we have XXY. This clearly shows when you do karyotyping where it allows you to separate the chromosomes and group them. And when you count them, when you organize after arranging them like that, so this would clearly tell you that there are 2 X chromosomes and 1 Y chromosome. This is in evidence. This is a diagnostic tool for chromosomal abnormalities. So karyotyping is a method.
So does this say that chromosome number 1 for everybody has this pattern, or for this particular human, this pattern is followed?
This is all this, except for this number, everything else more or less is same for humans. We all have more or less same sequences, 99.9% same sequences we have, similar sequences we have. So they show same pattern, doesn't matter whoever you take. With very slight variation that I don't think we can even observe that. Okay, notice that, but today technology might be able to capture it, but usually we get the same pattern. Because the density or not density, the ability of certain regions in our chromosomes to extract or take more stain is more or less preserved in humans. So this pattern can be seen. So pattern is not used for determining the problem. Pattern is only used for pairing them, for grouping them. Length and pattern is, so you notice that length is also different, right? All chromosomes are of same length in humans. If you see it, how are these organized? Do you see any pattern? I told you one, we grouped
reducing length.
Pattern. Reduction in length, decreased length. So these are the longest chromosomes. And as we go forward, the chromosome size is decreasing. 21, 22, 20, 23, sorry, 21, 22 are shortest. Some of the shortest chromosomes. Okay, based on length, we organize them. This is called karyotype. Karyotype. Okay, karyo means nucleus, karyo. Okay, right. So this is how we determine the chromosomal abnormality. This is a diagnosis. Is there any solution for disease management? Is there any disease management method? Can we manage this problem? Can we cure this problem? Can we treat it? Is there a solution to this problem?
Maybe if we can detect in the womb itself, probably.
Okay, we can detect in the womb. Okay. Are there methods to detect in the womb? We'll discuss it in 9.4D, genetic screening. We'll talk about in genetic screening, there are methods to detect the problem in mother's womb. And when do we suspect it? Everyone has to check for this? No. If there is a family history. That is where genetic counseling comes into the picture. Okay, in mother's womb. So what can you do if you identify it in mother's womb? Can you correct the problem? You only know that baby with this problem will be born. You have no way to cure this problem. We have no solution to this problem. If parents want, they can decide not to have such a baby. They can take an informed decision. If you see the laws that we have, even the recent amendment to the, what is that called? Oh, it means something. Genetics testing, pre-implantation genetic screening or pre-implantation genetic testing allows in certain cases where if there are rare abnormalities in our DNA, couple is allowed to go for abortion. Okay. So legally it is allowed in few such cases so that they can decide not to have a child. But otherwise there is no cure to this problem. But this problem can be managed. I'll give you one source on this, okay? This is a very good source that you can refer to where you'll find some additional information on this. So there is an American website, okay, which provides us enough information on this. So NIH, National Institute of Health related website that we have. So there's a good website for genetic abnormalities like that. NHS. Klinefelter syndrome. Genome. Look at this site. I'm going to share this link to you. These are the very authentic sources for this type of information, rather than maybe reading from Peanut. So this type of information is very, very useful to us. Chart box. I'm sending you a link on it. Okay, I'll show you this website as well. Use this type of. So this is U.S. government's research websites, where they actually organize information for the general public. So these are very good tools for you, and you will get enough ideas and more authentic ideas on this. You see it here. So this is a website, genome.gov. Genome.gov. So US government website, National Human Genome Research Institute is called. So belongs to NIH, National Institute of Health of Americas. So here, Klinefelter syndrome, you clearly see what is Klinefelter syndrome. Okay, the information I provided you here, written as XXY. Symptoms of Klinefelter syndrome. So these things, increased risk for breast cancer. I was telling you 2 types of cancers can be seen here. So males who have Klinefelter syndrome have following symptoms: small, firm testes, small penis, sparse pubic area, armpits and facial hair, enlarged breasts, so tall structure. So those things I've listed to you. How is Klinefelter syndrome diagnosed? Karyotype, chromosomal analysis of karyotype is used to confirm the diagnosis. Okay, how is karyotyping is done? We'll talk about that later. In addition to this, many things, like for example, physical characteristics, sperm counts, hormonal levels. So additionally, these are also used. Clinical features are also used. How is it treated? So as such, there is no cure to this. So there are ways to manage this. How? If testosterone, decrease in testosterone is causing the problem, then what is a good solution to this? When you diagnose the problem, when a child is born, give them testosterone injections. Hormonal therapies they are called. Hormonal therapy, or specifically testosterone therapy, can be used for this. So write this type of information. This is value addition to your answer. Whichever, there are, I think many books talk about this information. They would write about what is Klinefelter, what causes this, and features. But write how is it diagnosed and how is it treated or how is it managed. One, testosterone therapy. Okay, so testosterone therapy can address some of the issues. So infertility, how is infertility can be handled? Usually they are infertile, as I was talking to you. Very few cases with an extra X chromosome had children. Okay, so infertility specialist, fertility clinics may help them. Sometimes fertility clinics may help them, because all these things, the non-disjunction we are talking about is not a heritable problem. You may wonder if one has Klinefelters in the family, it runs in the families. It won't run in families. These chromosomal abnormalities don't run in families. They are random events. Randomly, maybe because of age factor, maybe because of some exposure to certain toxins, this may happen. So if someone has Klinefelters, their future generations, it doesn't increase the risk factor in future generations, because it's one random event of non-disjunction. Few things like age and those factors may influence it. Okay.
But sir, in earlier classes we talked about usually heritable changes happen in the sex chromosomes.
Correct.
This being
exactly. So there we were referring to genetic diseases which are caused due to mutations. Let's say in X chromosome and Y chromosome, if there is a mutation, mutation is passed through that X and Y chromosomal DNA.
okay.
Correct? But here, the problem is happening because it is not separating during cell. There is no problem, no mutation in the chromosome itself. But during the cell separation, chromosomal separation process, cell division process, and abnormalities happen. So here, the reason is the abnormality in separation, and it is not running in families. The abnormality can happen with anyone. Okay, if it is a genetic disease because of a mutation like hemophilia, sickle cell anemia, yes, you are absolutely right. But chromosomal abnormalities like this don't run in families. I'll give you a review to read. I think one scientist from, I'm not wrong, I don't know whether University of British Columbia or University of Victoria, from the western part of Canada, she wrote a review on chromosomal abnormalities. I give usually as a reading material for you. But of course, most of the information I'm giving you in the class, if you want some reading, that is a nice review that I'm going to share with you. They nicely explain in that. They also get into a little bit of details about mechanism behind abnormalities and things like that. And what percentage problem from males, what percentage problem from females, also they discussed. If you can digest that much of information, do it. Otherwise, the information I'm giving from the class is sufficient.
Can you also share the amendment or the law we just discussed?
Sure, I will share the PRS, okay, summary of that, yes, for sure.
Oh.
This came, I think, along with the recent surrogacy bill, along with that ART bill, they put an amendment on this PIBs. I picked up PIBs from PIBs. We normally teach it for science and technology classes. Okay. Okay. So in addition to this, testosterone therapy and also help with fertility specialists or infertility specialists, and then educational interventions. We talk about learning disabilities, correct? So those disabilities can be cured by proper training, education interventions, and strong social support, thereby optimizing the individual's potential in adulthood. So this is something you can write to strengthen your answers. Okay. This is how karyotype looks like. This is theoretical diagram. The one I've shown you is the real diagram. Real Klinefelter syndrome karyotype. This is theoretical diagram. 2 X chromosome, 1 Y chromosome. Okay, this is also a good site. So where you will see the frequency, causes. Okay, in the causes, they get into the details about meiosis, what stage of meiosis. That will be a little too much, so don't worry about that. Inheritance. So Klinefelter syndrome is not inherited. The addition of extra chromosome occurs during the formation of gametes in affected person. So error, this is because of error called non-disjunction. Okay. Sometimes if you come across this term, don't get confused. They say mosaic Klinefelters. So meaning combinations. Some cells have extra X chromosome, some cells have normal X chromosome. So that is combinations is called mosaicism, mosaic nature. Because some cells tend to get rid of the extra X chromosome. So some cells may carry normal number of chromosomes, some carry extra number of chromosomes. This is called mosaic Klinefelter. To come across, I don't think you need to mention that, but be familiar with this. Okay. Right. Good. So this is the structure. This is how we learn about the other abnormalities as well, other numerical abnormalities as well. Okay. Quickly, I'll talk to you about the next one. So we'll follow the same approach. Yeah, these are the bent fingers I was telling you. So they're bent like this. This is called clinodactyly. Turner syndrome. First, I'll start with the diagnosis part. What is the genotype of the Turner syndrome based on this image, based on this karyotype?
(45X not).
45X, correct? All of them are in pairs. Only thing is it looks weird, the real image, because what normally they do is they drop cells on a slide. And when slide actually drops, chromosomes are flexible, right? They don't actually stand straight like this. So they come in varieties of shapes. So it is big headache for these cytologists, we call them cytologists, okay, to find a good spread of chromosomes. So it requires a lot of expertise, very laborious process. Okay, so in this, the chromosomes are paired like this, as you see. Up to 22 chromosomes, they're all in pairs, only 2 chromosomes for each of them. But only in the sex chromosome, you have 1 X chromosome, no Y, no additional X chromosome. So this is 45, X. So this is Turner syndrome. Now following the same approach, I'll quickly tell you, it is identified by Henry Turner. He's a US endocrinologist. You don't have to write endocrinologist, just for your information. So it is named after Turner, who identified it. Incidence rate, I already told you, 1 in 2000 cases. We already talked about this. Okay, I'll share these slides with you. My handout will be these slides. That is how I organize all the information in one go and place here. 1 in 2000 cases. Okay, most of the cases, studies show that most of it because of the maternal X chromosome. Missing of the maternal X chromosome. 80%. You can ignore this. This is a little bit more specific. And mosaics are also seen. What does it mean? I just explained you about mosaicism or mosaics. If they say mosaics are seen, what does that mean? They carry few cells with X0, few cells with XX. Mosaic condition is also seen. So in a way, 45X0 is somewhat opposite to what we have seen in Klinefelter syndrome. If you see the features, this is just to make you remember a bit easily. Okay, so look at the features. What did we talk about in the Klinefelter syndrome? They're taller. They're usually taller. Correct? Here they are shorter. Absence there, additional X chromosome. Here, absence of extra X chromosome. So you see the opposite effects here. They're short. They tend to be short. These are females. First of all, they are females, right? No Y chromosome. So these are genetically females. You can write that point here. Genetically females. No Y chromosome or absence of Y chromosome. Short stature. They tend to be shorter. And what do you notice here? Primary amenorrhea. So amenorrhea refers to absence of menstruation. So these people don't show the onset of menstruation. Again, hormonal issues here. X chromosome is absent. One X chromosome is absent. So reduction in hormones related to the females. And primary, initially, even to begin with, menstruation doesn't begin in them, basically. That is what it means. Absence or onset of menstruation doesn't happen. Okay. So similar, these are the opposite characteristics. There are some similarities to what we have seen with the Klinefelters. What are they? Secondary sexual development is reduced or is absent. Not completely absent, but reduced. So we're talking about females. So you give specific examples of what type of secondary sexual characteristics are absent. I'll highlight few of these things here. Poorly developed breast. Poor breast development. This is one point you can make a note of. And rudimentary ovaries. Rudimentary ovaries. Poorly developed ovaries. There we talked about poorly developed testes. In Klinefelters here, rudimentary or poorly developed ovaries, poor breast development. Their breast development is present where it's supposed to be absent. Here, poor development. So this is also similar with respect to that, and poor ovary development. So poor ovary development means less estrogen, because ovaries are the sites where female hormones are produced. That is why primary amenorrhea and secondary sexual development is affected. Okay. Any other facial features or physical features that you see here? If you can remember some of them, you won't be able to write all of them. The list I've given here is sufficient. But if not, if you want fold of skins, they normally have folds, extra additional folds here. So this is one clinical features that people can observe. Short and foldings. Fold. Skin folds are noticed here. And also facial features. They also have some folds. We'll talk about this in the races, concept of human races. There you will notice some of the eye features actually look like epicanthic fold, we say. There is an additional fold on the eye. Such facial features are also seen here. I'm not covering all of them because you won't be able to fit them in the exam. So I'm just highlighting few important ones. So short stature and few sexual related characteristics, poor breast development, rudimentary ovaries, and absence of menstruation process. Failure in secondary sexual development, where you write about both of these. And similar to what we talked about in Klinefelters, infertility is seen in them. Infertile, they're also infertile. Number one, they also have reduced intelligence. So these points more or less repeat for many of these diseases. Infertility, reduced intelligence, and neck fold, I've already told you, redundant neck skin or fold on the skin. I've already told you this. Congenital heart problems. They are born, congenital means by birth. They have heart problems when they are born. Okay. For example, constriction in aorta. Aorta is the main artery which carries blood to various parts of the human body from heart. So constriction in aorta is seen. Instead of writing that specifically, you can write heart problems, congenital heart problems. Heart problems during birth can be seen. Okay, right. So if you can remember, as I told you in the previous case, osteoporosis, cancers, diabetes, the Klinefelters people are prone to. Similarly, yeah, increased risk for hypertension. high blood pressure, hypertension, autoimmune thyroid. Thyroid cases happen, but because of the autoimmunity, self-immunity, immune system itself is destroying thyroid hormone producing glands. Auto, self-immunity, leading to thyroid disease. Obesity is also seen as a problem. Hypertension, obesity, you can remember together, and autoimmune thyroid. Okay? So it makes them prone to certain diseases like hypertension, you can do. Okay. So this is a diagnosis. Now tell me, can you guess and tell me what would be the disease management part? Diagnosis is karyotyping. How can the disease managed? We cannot cure the problem. We can try to treat the problem. Can you suggest some solutions? Exactly what we did for Klinefelter's.
Estrogen injections.
Correct. Hormonal therapies. Next.
Next, early detection.
Okay, genetic screening in case if they want to avoid the child like this.
Fertility again, fertility centers.
Fertility centers, okay, right. Next.
Maybe cosmetic surgeries for that extra skin.
Exactly, very good. Cosmetic surgeries. So these things you can actually write yourself. Once you have the idea about what can be done and what cannot be done. So given the technology that we have today, cosmetic surgeries can be done. Fix this redundant neck skin, for example. Facial features can be corrected. Because socially, these can be considered as a problem, right? Because societal issues, this can be rectified. Congenital heart problems, surgeries can be done. When they identify the problem, constriction in the aorta can be fixed. For issues with intelligence, can you do something for this? Education intervention, training, additional training, education intervention to improve their IQ. Environmental approach. So we can actually use extra training to train them, to catch them with the children of the same age. Okay, that can be done. Right. So I'm stopping the session here. So in the next class, we are going to go through the remaining diseases. Now we got the structure for the component, right? So next class, we'll continue with the super females. And I'm also going to talk to you about intersex. Okay. So what causes that ambiguity in the gender determination? Why some people have both feminine and male characteristics, in addition to Klinefelters that we talked today. Plus, we move on to the autosomal diseases, Down syndrome, Patau's, Edward, Krejci-Chart syndrome. Hopefully, I'll be able to finish this in the next class, most of them. Okay, because now we have the structure. I'll just have to explain you the logic behind each of those characteristics. I'll do that. Then we'll move on to the 9. I think before going to 9.4D, I will talk to you about structural abnormalities and then genetic imprints. Many questions are coming from this. I'll briefly tell you a couple of questions that came, so you know what to expect in the exam from this part. Describe Turner syndrome and Klinefelter syndrome. 15 marker. Describe Turner and Klinefelter syndrome. Can you do that? Can you answer that question? Describing Turner and Klinefelter. This came in 2014. This question repeatedly comes, directly or indirectly. Like, I know even last year we had a question on this. Discuss chromosomal aberrations in man, illustrating with examples. More broader question. Discuss chromosomal aberrations or chromosomal abnormalities in man, illustrating with examples. So, what are chromosomal abnormalities in humans? Explain them with examples. We can use these type of examples for that. Such question can be written. What else? Turner's, Klinefelters. Yes. Question repeated again. Discuss chromosomal aberrations and manifestations of Klinefelter syndrome and Turner syndrome. These two most commonly come. Klinefelter, Turner's we discussed today, and Down syndrome are the most commonly asked questions from this unit. Okay, I gave you the subheadings to cover. Initial part, genotype you write, incidence rate you write, genotypically male or female you write. Correct? First part is that. Then you write the features with a rough diagram, and then diagnosis, management. Did you be able to cover all those aspects? Okay, good. Right. So we'll stop it here. Okay, right. So I'll continue in the next class.
Thank you, sir.
You're welcome. So happy Pongal, happy Lori, people who are celebrating today's festival. Okay, right. I think we happen to have a class today. Right. Bye, guys.
Thanks.